Chapter 5: DNA and Chromosomes. Life depends on the ability of cells to store, retrieve, and translate the genetic instructions required to make and maintain a living organism. This hereditary information is passed on from a cell to its daughter cells at cell division, and from generation to generation in multicellular organisms through the reproductive cells. These instructions are stored within every living cell in its genes, the information containing elements that determine the characteristics of a species as a whole and of the individuals within it. At the beginning of the 20th century, when genetics emerged as a science, scientists became intrigued by the chemical nature of genes. The information in genes is copied and transmitted from cell to daughter cells millions of times during the life of a multicellular organism, and it survives the process essentially unchanged. What kind of molecule could be capable of such accurate and almost unlimited replication, and also be able to direct the development of an organism and the daily life of a cell? What kind of instructions does the genetic information contain? How are these instructions physically organized so that the enormous amount of information required for the development and maintenance of even the simplest organism can be contained within the tiny space of a cell? The answers to some of these questions began to emerge in the 1940s, when it was discovered from studies in simple fungi that genetic information consists primarily of instructions for making proteins. Proteins are the macromolecules that perform most of the cell's functions. They serve as building blocks for cell structures. They form the enzymes that catalyze the cell's chemical reactions. They regulate gene expression, and they enable cells to move and to communicate with one another. With hindsight, it is hard to imagine what other type of instructions the genetic information could have contained. The other crucial advance made in the 1940s was the recognition that deoxyribonucleic acid, DNA, was the likely carrier of this genetic information. But the mechanism whereby the hereditary information is copied for transmission from cell to cell, and how proteins are specified by the instructions in the DNA, remained completely mysterious until 1953, when the structure of DNA was determined by James Watson and Francis Crick. The structure immediately revealed how DNA might be copied or replicated, and it provided the first clues about how a molecule of DNA might encode the instructions for making proteins. Today, the fact that DNA is the genetic material is so fundamental to biological thought that it is difficult to appreciate what an enormous intellectual gap this discovery filled. In this chapter, we begin by describing the structure of DNA. We see how, despite its chemical simplicity, the structure and chemical properties of DNA make it an ideal raw material for making genes. The genes of every cell on Earth are made of DNA, and insights into the relationship between DNA and genes have come from experiments in a wide variety of organisms. We then consider how genes and other important segments of DNA are arranged in the long molecules of DNA that are present in the chromosomes of cells. Finally, we discuss how eukaryotic cells fold these long DNA molecules into compact chromosomes that are contained inside the nucleus. This packing has to be done in an orderly fashion, so that the chromosomes can be replicated and appointed correctly between the two daughter cells at each cell division. It must also allow access of chromosomal DNA to enzymes that repair it when it is damaged, and to the specialized proteins that direct the expression of its many genes. This is the first of 5 chapters that deal with basic genetic mechanisms, the ways in which the cell replicates, repairs, expresses, and occasionally improves the genetic information carried in its DNA. The totality of this information in each cell is called its genome. In Chapter 6, we discuss the mechanisms by which the cell accurately replicates and repairs DNA. We also describe how DNA sequences can be rearranged through the process of genetic recombination. Gene expression, the process by which the information encoded in DNA is interpreted by the cell to guide the synthesis of proteins, is the main topic of Chapter 7. In Chapter 8, we describe how gene expression is controlled by the cell to ensure that each of the many thousands of proteins encrypted in its DNA is manufactured at the proper time and place in the life of the cell. We turn in Chapter 9 to a discussion of how present-day genes and genomes evolved from distant ancestors. An account of the experimental techniques used to study both DNA and its role in fundamental cellular processes will be presented in Chapter 10. The structure and function of DNA. Well before biologists understood the structure of DNA, they had recognized that inherited traits and the genes that determine them were associated with the chromosomes. Chromosomes, named from the Greek chroma, color, because of their staining properties, were discovered in the 19th century as thread-like structures in the nucleus of the eukaryotic cell that become visible as the cell begins to divide. Figure 5.1. As biochemical analysis became possible, researchers learned that chromosomes contain both DNA and protein. But which of these components encoded the organism's genetic information was not at all clear. We now know that the DNA carries the hereditary information of the cell, and that the protein components of chromosomes function largely to package and control the enormously long DNA molecules. But biologists in the 1940s had difficulty accepting DNA as a genetic material because of the apparent simplicity of its chemistry. See How We Know, pages 174 to 176. DNA was thought of as simply a long polymer composed of only 4 types of subunits, which are chemically very similar to each other. Then, early in the 1950s, DNA was examined by X-ray diffraction analysis, a technique for determining the three-dimensional atomic structure of a molecule, discussed in Chapter 4, pages 158 to 160. The early results indicated that DNA is composed of 2 strands wound into a helix. The observation that DNA is double-stranded was of crucial significance. It provided one of the major clues that led in 1953 to a correct model for the structure of DNA. As soon as the Watson-Crick model of DNA structure was proposed, DNA's potential for replication and information encoding became apparent. In this section, we examine the structure of the DNA molecule and explain in general terms how it is able to store hereditary information. A DNA molecule consists of 2 complementary chains of nucleotides. A molecule of deoxyribonucleic acid, DNA, consists of 2 long polynucleotide chains. Each of these DNA chains, or DNA strands, is composed of 4 types of nucleotide subunits, and the 2 chains are held together by hydrogen bonds between the base portions of the nucleotides, Figure 5.2 and see Panel 2.7, pages 76 to 77, for a description of hydrogen bonds. As we saw in Chapter 2, Panel 2.6, pages 74 to 75, nucleotides are composed of a 5-carbon sugar to which are attached 1 or more phosphate groups and a nitrogen-containing base. For the nucleotides in DNA, the sugar is deoxyribose attached to a single phosphate group, hence the name deoxyribonucleic acid. The base may be either adenine A, cytosine C, guanine G, or thymine T. The nucleotides are covalently linked together in a chain through the sugars and phosphates, which thus form a backbone of alternating sugar-phosphate. See Figure 5.2b. Because it is only the base that differs in each of the 4 types of subunits, each polynucleotide chain in DNA can be thought of as a necklace. The backbone strung with 4 types of beads, the 4 bases, A, C, G, and T. These same symbols, A, C, G, and T, are also commonly used to denote the 4 different nucleotides, that is, the bases with their attached sugar and phosphate groups. The way in which the nucleotide subunits are linked together gives a DNA strand a chemical polarity. If we imagine that each nucleotide has a knob, the phosphate, and a hole, see Figure 5.2a, each chain formed by interlocking knobs with holes, will have all of its subunits lined up in the same orientation. Moreover, the two ends of the chain can be easily distinguished, as one will have a hole, the 3' hydroxyl, and the other a knob, the 5' phosphate. This polarity in a DNA chain is indicated by referring to one end as the 3' end and the other as the 5' end. This convention is based on the details of the chemical linkage between the nucleotide subunits. The two polynucleotide chains in the DNA double helix are held together by hydrogen bonding between the bases on the different strands. All the bases are therefore on the inside of the helix, with the sugar phosphate backbones on the outside. See Figure 5.2d. The bases do not pair at random, however. A always pairs with T, and G always pairs with C. Figure 5.6. In each case, a bulkier 2-ring base, a purine, see panel 2.6, pages 74 to 75, is paired with a single ring base, a pyrimidine. Each purine-pyrimidine pair is called a base pair, and this complementary base pairing enables the base pairs to be packed in the energetically most favorable arrangement in the interior of the double helix. In this arrangement, each base pair is of similar width, thus holding the sugar phosphate backbones an equal distance apart along the DNA molecule. The members of each base pair can fit together within the double helix because the two strands of the helix run antiparallel to each other. That is, they are oriented with opposite polarities. See Figure 5.2c and d. The antiparallel sugar phosphate strands then twist around each other to form a double helix containing 10 base pairs per helical turn. Figure 5.7. This winding also contributes to the energetically favorable conformation of the DNA double helix. A consequence of the double helix base pairing requirements is that each strand of DNA molecule contains a sequence of nucleotides that is exactly complementary to the nucleotide sequence of its partner strand. An A always matches a T on the opposite strand, and a C always matches a G. This complementarity is of crucial importance when it comes to both copying and repairing the DNA, as we discuss in Chapter 6. An animated version of DNA structure can be seen in Movie 5.1. The structure of DNA provides a mechanism for heredity. Genes carry biological information that must be copied and transmitted accurately when a cell divides to form two daughter cells. This situation poses two central biological problems. How can the information for specifying an organism be carried in chemical form? And how is it accurately copied? The discovery of the structure of the DNA double helix was a landmark in 20th century biology because it immediately suggested answers to these two questions and thereby resolved the problem of heredity at the molecular level. In this chapter, we outline the answer to the first question. In the next chapter, we address in detail the answer to the second. DNA encodes information in the order or sequence of the nucleotides along each strand. Each base, A, C, T, or G, can be considered as a letter in a 4-letter alphabet that is used to spell out biological messages in the chemical structure of the DNA. Figure 5.8. Organisms differ from one another because their respective DNA molecules have different nucleotide sequences, and consequently carry different biological messages. But how is the nucleotide alphabet used to make up messages, and what do they spell out? It had already been established some time before the structure of DNA was determined that genes contain the instructions for producing proteins. The DNA messages, therefore, must somehow encode proteins. Consideration of the chemical character of proteins makes the problem easier to define. As discussed in Chapter 4, the function of a protein is determined by its three-dimensional structure, and its structure in turn is determined by the sequence of the amino acids in its polypeptide chain. The linear sequence of nucleotides in a gene must therefore somehow spell out the linear sequence of amino acids in a protein. The exact correspondence between the 4-letter nucleotide alphabet of DNA and the 20-letter amino acid alphabet of proteins, the genetic code, is not obvious from the structure of the DNA molecule, and it took more than a decade after the discovery of the double helix to work it out. In Chapter 7, we describe this code in detail in the course of explaining the process, known as gene expression, through which a cell transcribes a nucleotide sequence of a gene into the nucleotide sequence of an RNA molecule, and then translates that information into the amino acid sequence of a protein. Figure 5.9. The complete set of information in an organism's DNA is called its genome. The term is also used to refer to the DNA that carries this information. The total amount of this information is staggering. Written out in the 4-letter nucleotide alphabet, the nucleotide sequence of a very small gene from humans occupies a quarter of a page of text, while the complete sequence of the human genome would fill more than 1,000 books the size of this one. Herein lies a problem that affects the architecture of all eukaryotic chromosomes. How can all this information be packed neatly into every cell nucleus? In the remainder of this chapter, we discuss the answer to this question. The structure of eukaryotic chromosomes. Large amounts of DNA are required to encode all the information needed to make even a single-celled bacterium, and far more DNA is needed to encode the instructions for the development of multicellular organisms like ourselves. Each human cell contains about 2 meters of DNA, yet the cell nucleus is only 5 to 8 micrometers in diameter. Tucking all this material into such a small space is the equivalent of trying to fold 40 kilometers, 24 miles of extremely fine thread into a tennis ball. In eukaryotic cells, very long double-stranded DNA molecules are packaged into structures called chromosomes, which not only fit readily inside the nucleus, but can be easily apportioned between the 2 daughter cells at each cell division. The complex task of packaging DNA is accomplished by specialized proteins that bind to and fold the DNA, generating a series of coils and loops that provide increasingly higher levels of organization and prevent the DNA from becoming an unmanageable tangle. Amazingly, the DNA is compacted in a way that allows it to remain accessible to all of the enzymes and other proteins that replicate it, repair it, and direct the expression of its genes. Bacteria typically carry their genes on a single circular DNA molecule. This molecule is also associated with proteins that condense DNA, but these proteins differ from the ones that package eukaryotic DNA. Although this prokaryotic DNA is called a bacterial chromosome, it does not have the same structure as eukaryotic chromosomes, and less is known about how it is packaged. Our discussion of chromosome structure in this chapter will therefore focus entirely on eukaryotic chromosomes. Eukaryotic DNA is packaged into multiple chromosomes. In eukaryotes, such as ourselves, the DNA in the nucleus is distributed among a set of different chromosomes. The human genome, for example, contains approximately 3.2 times 10 to the power of 9 nucleotides parceled out into 24 chromosomes. Each chromosome consists of a single, enormously long linear DNA molecule associated with proteins that fold and pack the fine thread of DNA into a more compact structure. The complex of DNA and protein is called chromatin. In addition to the proteins involved in packaging the DNA, chromosomes are also associated with many other proteins involved in gene expression, DNA replication, and DNA repair. With the exception of the germ cells, sperm and eggs, and highly specialized cells that lack DNA entirely, such as the mature red blood cell, human cells each contain 2 copies of each chromosome, one inherited from the mother and one from the father. The maternal and paternal chromosomes of a pair are called homologous chromosomes, homologs. The only non-homologous chromosome pairs are the sex chromosomes in males, where a Y chromosome is inherited from the father and an X chromosome from the mother. In addition to being different sizes, human chromosomes can be distinguished from one another by a variety of techniques. Each chromosome can be painted a different color, using sets of chromosome-specific DNA molecules coupled to different fluorescent dyes. Figure 510. This involves the technique of DNA hybridization, which will be described in detail in Chapter 10. A more traditional way of distinguishing one chromosome from another is to stain the chromosomes with dyes that bind to certain types of DNA sequences. These dyes mainly distinguish between DNA that is rich in AT nucleotide pairs and DNA that is GC rich, and they produce a striking and reliable pattern of bands along each chromosome. Figure 511. The pattern of bands on each type of chromosome is unique, allowing each chromosome to be identified and numbered. A display of the full set of 46 human chromosomes is called a human karyotype. If parts of a chromosome are lost or switched between chromosomes, these changes can be detected by changes in the banding patterns. Cytogeneticists use alterations in banding patterns to detect chromosomal abnormalities that are associated with some inherited defects and with certain types of cancer. Chromosomes contain long strings of genes. The most important function of chromosomes is to carry the genes, the functional units of heredity. Figure 513. A gene is usually defined as a segment of DNA that contains the instructions for making a particular protein, or in some cases, a set of closely related proteins. Although this definition fits the majority of genes, some genes direct the production of an RNA molecule instead of a protein as their final product. Like proteins, these RNA molecules perform a diverse set of structural and catalytic functions in the cell, as we will see in later chapters. As might be expected, some correlation exists between the complexity of an organism and the number of genes in its genome. For example, the total number of genes ranges from less than 500 for a simple bacterium to about 25,000 for humans. Bacteria and some single-celled eukaryotes have especially compact genomes. The DNA molecules that make up their chromosomes are little more than strings of closely packed genes. However, chromosomes from many eukaryotes, including humans, contain, in addition to genes, a large excess of interspersed DNA, the majority of which does not seem to carry critical information. This DNA is sometimes called junk DNA because its usefulness to the cell has not yet been clearly demonstrated. Although the particular nucleotide sequence of most of this DNA might not be important, the DNA itself, acting as spacer material, may be crucial for the long-term evolution of the species and for the proper activity of genes. In addition, comparisons of the genome sequences from many different species reveals that a portion of this extra DNA is highly conserved among related species, indicating that it serves an important function, although we don't yet know what it is. We take up this question in greater detail in Chapter 9. In general, the more complex an organism, the larger is its genome. But this relationship does not always hold true. The human genome, for example, is 200 times larger than that of the yeast, as Cerevisiae, but 30 times smaller than that of some plants, and at least 60 times smaller than some species of amoeba. Furthermore, how the DNA is apportioned over chromosomes also differs from one species to another. Humans have 46 chromosomes, but a species of small deer has only 6, while some carp species have more than 100. Even closely related species with similar genome sizes can have very different numbers and sizes of chromosomes. Figure 514. Thus, although gene number is roughly correlated with species complexity, there is no simple relationship between gene number, chromosome number, and total genome size. The genomes and chromosomes of modern species have each been shaped by a unique history of seemingly random genetic events, acted on by specific selection pressures. Chromosomes exist in different states throughout the life of a cell. To form a functional chromosome, a DNA molecule must do more than simply carry genes. It must be able to be replicated, and the replicated copies must be separated and partitioned reliably into daughter cells at each cell division. These processes occur through an ordered series of events, known collectively as the cell cycle. This cycle of cell growth and division is briefly summarized in Figure 5.15 and will be discussed in detail in Chapter 18. Only two broad stages of the cell cycle need concern us in this chapter: interphase, when chromosomes are duplicated, and mitosis, when they are distributed to the two daughter nuclei. During interphase, the chromosomes are extended as long, thin, tangled threads of DNA in the nucleus and cannot be easily distinguished in the light microscope. We refer to chromosomes in this extended state as interphase chromosomes. Specialized DNA sequences found in all eukaryotes ensure that the interphase chromosomes replicate efficiently. Figure 5.16. One type of nucleotide sequence acts as a replication origin, where duplication of the DNA begins. As we discuss in Chapter 6, eukaryotic chromosomes contain many replication origins to ensure that the entire chromosome can be replicated rapidly. Another DNA sequence forms the telomeres, found at each of the two ends of a chromosome. Telomeres contain repeated nucleotide sequences that enable the ends of chromosomes to be replicated, as we discuss in Chapter 6. They also cap the end of the chromosome, preventing it from being mistaken by the cell as a broken DNA molecule in need of repair. When the cell cycle reaches M phase, the DNA coils up, adopting a more and more compact structure, ultimately forming highly compacted or condensed mitotic chromosomes. This is a state in which chromosomes are most easily visualized. In this highly condensed state, duplicated chromosomes can be readily separated when the cell divides. See Figure 5.16. Once the chromosomes have been condensed, it is the presence of the third specialized DNA sequence, the centromere, that allows one copy of each duplicated chromosome to be apportioned to each daughter cell. Figure 5.17. We describe the central role that centromeres play in cell division in Chapter 18. Interphase chromosomes are organized within the nucleus. The nucleus is surrounded by a nuclear envelope formed by two concentric membranes. The nuclear envelope is punctured at intervals by nuclear pores, which actively transport selected molecules to and from the cytosol, described in detail in Chapter 15, and is supported by the nuclear lamina, a network of protein filaments that forms a thin layer underlying and the inner nuclear membrane, discussed in Chapter 17. Inside the nucleus, the interphase chromosomes, although longer and finer than mitotic chromosomes, are nonetheless organized in various ways. First, each interphase chromosome tends to occupy a particular region of the nucleus, and so different chromosomes do not become extensively entangled with one another. Figure 5.18. In addition, specific regions of chromosomes are attached to sites on the nuclear envelope or the nuclear lamina. The most obvious example of chromosome organization in the interphase nucleus is the nucleolus. Figure 5.19. The nucleolus is where the parts of the different chromosomes carrying genes for ribosomal RNA, the red knobs in Figure 5.11, cluster together. Here, ribosomal RNAs are synthesized and combined with proteins to form ribosomes, the cell's protein synthesizing machines, discussed in Chapter 7. The DNA in chromosomes is highly condensed. As we have seen, all eukaryotic cells, whether in interphase or mitosis, package their DNA tightly into chromosomes. Human chromosome 22, for example, contains about 48 million nucleotide pairs, stretched out end to end. its DNA would extend about 1.5 centimeters. Yet, during mitosis, chromosome 22 measures only about 2 micrometers in length, that is, nearly 10,000 times more compact than the DNA in its extended form. This remarkable feat of compression is performed by proteins that coil and fold the DNA into higher and higher levels of organization. The DNA of interphase chromosomes, although less condensed than that of mitotic chromosomes, figure 520, is still packaged tightly, with a compaction ratio of about 500-fold. In the next sections, we introduce the specialized proteins that make this compression possible. Bear in mind, though, that chromosome structure is dynamic. Not only do chromosomes condense and relax in concert with the cell cycle, but different regions of the interphase chromosome must unpack to allow cells to access specific DNA sequences for replication, repair, or gene expression. Chromosome packaging must be flexible enough to allow rapid, localized, on-demand access to the DNA. Nucleosomes are the basic units of eukaryotic chromosome structure. The proteins that bind to the DNA to form eukaryotic chromosomes are traditionally divided into two general classes, the histones and the non-histone chromosomal proteins. Histones are present in enormous quantities, more than 60 million molecules of several different types in each cell, and their total mass in chromosomes is about equal to that of the DNA itself. The complex of both classes of protein with nuclear DNA is called chromatin. Histones are responsible for the first and most fundamental level of chromatin packing, the nucleosome, which was discovered in 1974, when interphase nuclei are broken open very gently and their contents examined under the electron microscope. Most of the chromatin is in the form of fibers, each with a diameter of about 30 nanometers. Figure 521a. If this chromatin is subjected to treatment that causes it to unfold partially, it can then be seen under the electron microscope as a series of beads on a string. Figure 521b. The string is DNA, and each bead is a nucleosome core particle that consists of DNA wound around a core of proteins formed from histones.